My name is Gretchen Meyer, and I work with Dr. Lieber in skeletal muscle physiology. And the topic of this poster and my current research is the mathematical modeling of the function of Desmin in skeletal muscle. Desmin is a member of the intermediate filament protein family, another member of which, dystrophin, you might be more familiar with. Point mutations in dystrophin cause the muscular dystrophies. Similarly, point mutations in Desmin cause disorders called Desminopathies, which are less common but are still degenerative muscle disorders that eventually result in cardiac and respiratory arrest. So this is a very important line of research that we're very interested in. We have taken the approach here to model a putative function of Desmin and to see if the simulation results might correlate with what is seen physiologically in specifically mice that have been engineered to lack Desmin. So our putative function is that Desmin acts as a mechanical link in between myofibrils, which is depicted here in the schematic where Desmin is shown as yellow filaments surrounding Z-discs. It sort of acts as a scaffold to organize myofibrils and to align sarcomeres. In this figure here, you can see actual real fibers, which have been, their Z-discs have been labeled with GFP. And in the normal fibers, you can see relatively parallel straight lines indicating that the sarcomeres are aligned. Whereas in the knockout, the line, you can't really discern any kind of line. There's a lot of shear and misalignment between those Z-discs. So in our model, we sought to model each sarcomere as a specific element and to combine those elements into a finite element model. So we modeled each sarcomere as having a contractile element representing the myosin heads causing contraction in the sarcomere, and in parallel with a viscoelastic element which models the passive properties of the sarcomere. Each sarcomere is joined to its neighbor longitudinally to form myofibrils, which are shown here in the schematic. And then each myofibril is in parallel with another to form the whole fiber. The array that we used was actually 10 in parallel by 200 in series. And then each Z-disc at each element is linked to its neighbor laterally by a Desmin element, which is modeled as a linear spring. The spring constant for this element we took from the literature, as well as 17 other parameters from the model. So we really tried to focus the model on what is physiologically true about a real fiber. So we did two different kinds of simulations to investigate this model. The first was to simulate the fiber stretching passively. So essentially we just grabbed onto the ends and pulled. And we found that the alignment of Z-discs, as measured as the difference between the position of two adjacent Z-discs, was increased if the fiber lacked Desmin. So that's shown here in A, where the y-axis shows misalignment and the x-axis shows increasing stretch. So what we see is in pink, the fiber lacking Desmin has much greater misalignment than the normal fiber, and that misalignment increases with increasing stretch. The picture next to it is actually a quantification of the experimental data that I talked about over here, and you can see that it shows a similar trend where the fiber lacking Desmin has an increased misalignment, and that misalignment increases with increasing stretch. So that's good. It seems like that this is a reasonable function of Desmin acting as a link between Z-discs. Secondly, we also simulated isometric stress, which is the stress production when the fiber contracts at fixed length. And we saw that the Desmin null fiber, which is shown here in red, actually has less stress production than the normal wild-type fiber shown in blue, which has also been documented in the literature. Interestingly, though, we varied the Desmin concentration in the fiber as well as its location within the fiber. So we found that as you increase Desmin concentration, you got this sort of nonlinear logarithmic type curve. And this was especially interesting because there is a study that was done by another girl in my lab where she showed that the injury resulting from eccentric contraction, which can be correlated to initial stress production by a muscle, actually has that same type of logarithmic curve. Also interestingly, we found that if we localize Desmin to the exterior of the fiber, where the membrane and the extracellular matrix are, we actually found a much greater increase in stress production for a given concentration relative to if it had been distributed randomly or focused in the center of the fiber. So this is where we plan to concentrate our future work in this model, maybe trying to design some experiments to see if there is actually increased localization of Desmin at the fiber membrane.